All right, so thank you for inviting me to review with you the research that we have been doing with the CHAMP1 Foundation, Family Foundation at Children's Hospital of Philadelphia. So this is actually a program that we started working on probably, Jeff, about 5 years ago. What I wanted to go through, and I will try to do this relatively swiftly, is to tell you a little bit about the Center for Applied Genomics, because I mean we sort of have an infrastructure in place that is very well suited to take on a project like this, and then give you an overview of the CHAMP1 research and tell you sort of a little bit about the status of the gene therapy that we are developing. And then Thais, our postdoc who is working on this project, will present the mouse model that we are developing for gene therapy. But I wanted to start with this slide here because it's exactly 10 years now, or a decade, since the first sort of blueprint from a fetus was built using parental genome. So this was basically obtaining cell-free DNA from a blood of a pregnant mother and reconstructing the fetal genome. And what this allows, and obviously will for the future, is to basically diagnose any known gene mutation that we know of and potentially intervene. And we are doing this in animal models today. We are using this technology called CRISPR-Cas to replace defective genes with healthy genes. And this is really going to be the future of medicine once all the ethical issues around this technology is fully sorted out. Now, we heard from several presentations earlier in terms of the impact and role of induced pluripotent stem cells and stem cells themselves that look really promising as a potential therapeutic method or an approach to multiple different diseases, and this could become one of them, CHAMP1. CHOP was actually the first institute to get a gene therapy program approved in the United States. This was for Leber congenital amaurosis, so this is congenital blindness, where this adeno-associated viral vector was used to inject a normal gene into children who had mutations, so they were blind, and they gained vision again. Similarly, CAR T-cell therapy was CHOP and Penn were the first institute to institute that therapy, and it's now expanding beyond hematological cancer into multiple different other areas. But CRISPR-Cas9, the gene editing, is really going to be the future of medicine because this allows for so many different sort of potentials going forward. And getting this information now down to a single cell level that we can with the modern sequencing technology is really, really sort of the state of the art today in terms of knowledge and data granularity and understanding sort of what the mechanisms involved. Now, just very, very briefly, the Center for Applied Genomics was founded in 2006, and it's a really high throughput sort of genotyping and sequencing center and biobanking. So we've been collecting, we actually have about 600,000 unique samples from over 600,000 unique individuals there today. And we focus on all conditions, both common and rare, but rare diseases are sort of really our primary focus. And just to point out, we have a 26,000, almost 7 rare disease samples. About half of them have been resolved, but half of them are unresolved. But look, we also have a lot of autism and developmental disorder samples, patient samples in our biobank. And this becomes very important when you are sort of, need to compare conditions sort of such as CHAMP1 with other disorders. Many of these other disorders are polygenic, so it means that there are multiple genes. But when you have a well-characterized and well-defined disorder such as CHAMP1, that can be immensely informative to inform on how to deal with other perhaps much more common conditions. So not only can you benefit CHAMP1 patients, but you may be able to benefit multiple other patients. And I just wanted to emphasize that the biobank that we have automated robotic-based biobank systems, so all the samples that we collect here and by anyone are very safe and secure in that setting. Now, we have, through the years, since we founded CAG in 2006, really had multiple big milestone in terms of developing new therapeutic programs, and the hope is that this is going to be another one that we succeed with. I just wanted to mention one, which when we discovered the mutations in a gene called ALK, or anaplastic lymphoma kinase, in patients who have a rare pediatric cancer, which is called neuroblastoma. So when we found those mutations, and these are individuals who were born with these mutations, it subsequently turned out that patients with lung cancer, ovarian cancer, colon cancer, many other cancers, about 15 to 20% of these categories, they had the exact same mutations. So here we identified a mutation in patients that was like 1 in a million, and they informed on therapy of hundreds of thousands of patients worldwide. So it's quite remarkable what can be done sometimes with genetics. And who would have thought that patients with neuroblastoma, who are 1-year-old kids, would have something in common with a 70-year-old person who was a smoker with the same mutation. The difference being it was acquired mutation in the adult. It was a germline mutation that the child was born with in the child. So it's actually quite compelling and interesting. And so just to sort of, in terms of the start of this program, as I said, I was introduced to Jeff about 5-6 years ago. This was really just shortly after the gene was identified. And we sort of, really partnered on this to take on to try to resolve this disease to the extent we could. And what we did, for example, was to send our team from Philadelphia down to the first inaugural meeting, scientific family engagement meeting, as we have here now. This is the second meeting, to collect blood samples. And we collected blood samples, we isolated them, we confirmed the mutations in all of the individuals. We got a significant amount of data associated with this. We processed the cells, we isolated, we sent them to the Simon Foundation after full consent and permission. And these iPS cells were generated that were showed earlier this morning. And these will now be a foundation, because it's going to be critically important to understand if we have a successful gene therapy, you want to test these cells for these vectors, because there could be quite a difference in terms of how different mutations respond. So it's really, really important in that venue. And we have now already sort of established a mouse that we brought from the UC Davis to CHOP. We have developed plasmid and vector for this gene therapy treatment. And we partnered with, and Jeff drove this with a company called ViralGen, which is actually going to give us sufficient amount of vector to do these first proof of concept experiments in the mice. And so the mouse came from UC Davis, as I mentioned. This was Dr. Kristen Grimsrud, who generously shipped those mice over to us. It's a heterozygous mouse. And because of the background this mouse is based on, which is BLAC6 and C57, it's a very homogeneous, stable mouse. So any sort of mutation defect you introduce, that's really the only deviation. And so this mouse actually has a significant phenotype that I will show you shortly. And we are basically working now towards sort of establishing this as a model. And as I said, we partnered with ViralGen, which is capable to sort of take this from the mouse to the human studies. They're a GMP vector facility. And we are currently working towards sort of optimizing the phenotype readout so we can understand the rescue that we gain with the vector in the mouse, and similarly doing toxicology studies to establish that. Now, this slide was really covered by Koso and Alan this morning. This shows the mutations and shows the interactors of CHAMP1, so I'm not going to spend time on that. Just want to emphasize here that I believe that this condition might be significantly underdiagnosed because companies like GeneGX and Embry Genetics, when they looked into their genomic data and they published this, among 62,000 samples that were on exome at GeneGX, 22 of them had CHAMP1. And the problem is that many of the panels that are often being run to diagnose intellectual deficit conditions, it doesn't have the CHAMP1 gene on it, so therefore it's missed. But if you do an exome, you get the whole coding sequence of the genome. You can find them. So this is going to change and gonna improve. Now, this paper came from the Buxbaum's laboratory just very recently this year, and shows that there are still new mutations being identified. I'm sure that he's gonna, and his team gonna talk about this paper from Cold Spring Harbor similarly, just to validate that almost all of these mutations are always de novo, so they are not inherited, they are de novo mutations, and almost always nonsense, and some few are frame-shaped mutations. Similarly, this was well covered this morning by Alan and others. Obviously, the chromosomal regulatory segregation is critical, and these binding partners of Rev7 and other genes are similarly critical for to promote homologous recombination as a DNA repair mechanism. So the gene is clearly more important than we thought just a couple of years ago. Now, this is the phenotype. This sort of, one of the phenotypes you see in the mouse. You can see there's a box plot and scatter plot here on the left and right. This is the UC Davis mouse, and it's generated sort of using an endonuclease technology to basically just introduce a loss of function in the CHAMP1 gene. And you can see that there's a very different sort of, this is really measuring movements, if you will. There's a very significant difference between the wild type and the knockout. And here are sort of the most critical parts of the phenotypes on the initial phenotyping that was done by Dr. Grimsrud at UC Davis. And we are repeating all of this here now at CHOP. So dysmorphic features are seen, stunted growth, altered mentation, increased sort of startle response or anxiety type behavior, and autistic-like behavior in terms of the isolation and how the mice interact with other mice. So these are significant sort of variables. And it doesn't matter if they are exactly the same as the human, if they are significant enough so you can measure them consistently and then give a gene therapy and see that you reverse them, you rescue them. That's all we need to know. And the adeno-associated viral vector that we are using, so this is a modified virus, so it cannot cause a human disease. So it's a safe approach from that standpoint. And we can control exactly the load that we place into the human being with this, either intravenously or intrathecally, if this is given, and we are going to sort of pursue both approaches. And so the objective here, we envision that this may be an effective way of moving forward, but this is basically to evaluate if we can rescue any of these phenotype stages that we have seen, and then to do a very comprehensive toxicology exposure in the mice to ensure that we are safe with the type of vector that we are using. And when we look at the efficacy point, it's going to be critically important to investigate sort of the timing of the intervention. So that's going to be one of the main focus in this study. So there are different sort of stages. We don't necessarily have to go through this, but we are going to create also inducible mouse, so we can induce the knockout at any time and give therapy to better understand the relationship. The transduction studies, I'll show you that on the next slide. The functional assessment, similarly, that we do, and so it's really important to treat the wild type mice also with this viral vector, and we have the viral vector now to do that because you need to understand whether there are sort of off-target effect if you, for example, dose the wild-type mouse with this vector. Is that tolerated? Is it safe? And then, of course, we dose the mutant to ensure that we get rescue of the phenotype. And how you titrate this is obviously going to be sort of, monitored in a very systematic and close way. And we will start the titration initially probably around 1 week, and then go to 2, 3, 4, 5, 6, etc. The ability here to generate and rescue this in the mice, we believe is significant and likely to occur. And then we basically use disconventional methods to assess the expressivity of this in the therapy with regular sort of protein and immunohistochemistry and PCR techniques. Now, these are sort of, the most critical, of course, human symptoms. Not all of them can be assessed in the mouse, but that is not really necessarily critical. If we can assess some of them, we are assessing basically whether the dysfunction from loss of function of this gene in the human body, in the mouse organism, can be rescued, and many of the phenotypes in green are actually overlapping with what we see, and I showed you on the slide before. And so after we establish the safe and effective therapy in the mouse, the goal would be to subsequently move this into human. We have a very excellent experience with this at CHOP. We are using this vector to treat multiple different disorders, thalassemia, sickle cell anemia, and multiple other conditions. And so this is sort of done in a very stringent and careful way, obviously all regulated by IRB and FDA. And again, critical time is obviously a key factor. Dose of recombinant gene necessary in a haploinsufficient individual obviously will be critical, and all of this sort of comparative measures will be worked out. And as I mentioned, resolving this condition could inform on multiple other conditions that could sort of scale and extend beyond the benefits of just these individuals. So Thais Carneiro, who is a postdoc who basically moved specifically from Brazil to take on this role, is now going to show you sort of how she is working with the mouse and the cell-based studies that we are doing to address these sort of questions that I raised. And with that, I will stop and thank you. And there is an army of people to thank, of course, listing specifically the recruitment team, and there are a couple of the recruitment members here now to draw blood if any of the families are willing to participate. And Thais, Marina, and Brian Stanovsky, they are sort of the lead people working on this program. And since Abraham Lincoln said, the best way to predict the future is to create it, no one is going to resolve this problem for us. We have to do it ourselves. And here we are to do that. So thank you all, and happy to take questions.
Second mic.
Oh, no.
Wait.
Oh, here. Maybe just in the interim, I put those slides here because, interestingly, Iceland has only, where I'm from, has only 350,000 people, and there is a patient there who was just diagnosed with CHAMP1. It's a deletion, and so this is the time to visit. So maybe we'll have this meeting in Iceland at some point because the Northern Lights, or if we do it in the summer, it's also beautiful. So.
I have a quick question for you over here. So it might have been on purpose, but can you comment on the capsid being used or the area of the brain you're targeting or looking to target?
Well,
if that was intentionally left out, that's okay too.
Yeah, I don't think that we necessarily, because I mean, if we administer the vector intrathecally, it may have to be the administration route. We obviously sort of, don't necessarily control exactly where that vector goes. But the interesting thing, I didn't emphasize that perhaps enough, is that the mouse model from UC Davis has significant CT and MRI abnormalities. So there is a way to potentially monitor brain rescue as well in these mice. But where exactly, as I said, I mean that obviously sort of depends on where these neurons are, where this gene is most relevant, whether we are successful or not.
Would you consider ICM?
We could, absolutely.
Penn and Chap have a lot of experience. I used to be
Yeah.
at Penn, so
Okay.
I know a lot about the ICM programs there, and it might be something to consider.
Yeah, I mean, obviously there are many more better gene therapy experts there that I have access to, and so this project is really being done sort of with a lot of knowledgeable people in the field, probably some of the most knowledgeable people in the field there is, and so we have a lot of guidance and advice for this.
Just out of curiosity, when do you have a timeline for using the vector in the mouse model?
So the mouse is basically, Thais is actually going to talk about that, Jeff, so maybe I'll leave it to her to tell.
Are there any other questions?
Wait, so there's going to be a follow-up? Wait, just ask a quick question. So that was beautiful presentation. You also want to just show that the AAV vector corrects the cells as well, because we're assuming this is a haploinsufficiency disease. But you see that the truncated protein is highly expressed in a lot of the cell lines that we've been looking at. And I'm just wondering if overexpressing you may need to overexpress the recombinant protein or any thoughts about then moving, of course, going to the mouse models at the same time makes good sense, but I just worry about just the cell lines. Can you correct them?
Sure, and that's why I think similarly the studying the iPS cells from the individual mutants is going to be very, very valuable in that context to see measurable functionality of the cell sort of output, whether we can change that with vector. And so, but it's a great point, and certainly something we have to address. Stefano?
Yeah, just like another question. Does this vector pass through the blood-brain barrier like easily, or they are like, you find some?
So that looks like it does. So we are going to see what we accomplish intravenously, but we will very likely, need intrathecal administration. Yeah.